nice introduction. Okay. Yes, I'm just confirming the recording. I'm ready to start, and I would like to say that if students want to ask any questions, you can interrupt me. Just turn on your microphone and ask something. It's totally okay to talk during the lecture, so I can always go back to the slides. It's fine. Okay, today I'm going to talk about the clonal biology, right, and how systematic clonal reconstruction at single cell level helps us to understand embryonic development. In general, embryonic development is probably the most interesting part of modern biology because through this, we learn how the whole multicellular body with all diversity of cell types arises from a single cell, which is a zygote, right? Basically, we come from a single cell, and that single cell originates from gametes from our parents. And starting from a single cell, we create the entire diversity of all cell types performing different functions in our body, being properly positioned, connected, functionally integrated, right? So there is an extraordinary complexity in this process. I think it's a fun time to be developmental biologist. So, and the clonal information gives you this level of understanding when you can actually track the families of cells and see which cell comes from which cell and how they diversify, right? Like yellow cells or red cells, they can be families, they can be clones, and these clones are different. And when we learn about these differences and what is driving them, we get completely new information about the process of embryonic development. Helps us to really understand it at deeper level. So it's kind of an instrument, a window into development, this type of clonal information. And but there is a problem, of course, in the field, the historical problem. Traditionally, people looked just at paths, how cells basically undergo these progressive fate restrictions, and they were collecting clonal data to understand how the fates are generally partitioned in developing embryo, and if we have the time axis, which is here, right? So this will be kind of like process going on and on and on until the whole diversity of cell types is created. So basically, the whole development is approximated like this graph, right? So this is a very intuitive way of depicting the dynamics of development. However, this is very far from reality because in reality, every cell type is created at certain proportions, right? And every decision in progenitor cells is biased, and it's basically this bias defines what will be that proportion. This proportion is very important because when we construct the body from many units, we need to produce them at the right numbers in the right places, right? So we have very dynamic system that is sensitive to signals that can kind of tune it, and that accounts for robustness of development. So we would like to get a little bit better way of looking at development. We want to assess collectives of individual progenitors. We want to be able to study biased fate decisions. And for this, we must collect the clonal information from thousands of individual progenitors, giving rise to different clones in an embryo. We don't want to have some kind of very generalized approximation, what we have on the left. We want to have it very specific now. And the thing is that even similarly looking progenitors, basically, or same type of progenitors, like neuroprogenitors in the early nervous system, or mesodermal progenitors, right, or neurocrust cells, they would do slightly different things in different parts of the body because they will be subjected to different signals, different signaling cues. So this cell will generate a lot of A fate and a little bit of B fate and no C fate and no D fate. And the same progenitor, just in a different position, will generate A, C, and D, and another one will generate B, C, and D. We can, of course, like get some superposition of all these progenitors and put them into this overarching tree, which is more informative. It collects the information about the proportions. But because every individual progenitor will do slightly different things, we call it the collective multipotency. And this is a very interesting term that basically tells us that embryonic development is achieved and the whole general multipotency is achieved as a sum of individual multipotencies. And these individual multipotencies, they go through the specific realization. Every individual progenitor will undergo this very specific realization of its own multipotency, and that will not cover the entire multipotency range. So multipotency is the potential, and every progenitor decides based on the signals and intrinsic information how to realize that potential fully or not fully, right? In principle, we can say that every human being, every person is extraordinarily multi-potent in terms of potential jobs and functions in the society. Indeed, we can take on any job in the society. We can be better or worse at it, but we can do anything that society needs. And yet, small babies, they grow up, they go through school, university, and typically we assume one profession where we kind of achieve the proficiency and we perform one major function in the society. So that's how our multipotency is channeled into concrete realization. And because embryos can consist of so many cells, so many progenitors build them, these progenitors also develop some mechanisms for crosstalks, and they will manage this type of realization strategy. And that's what we want to study using clonal information. So which cells, which progenitors are the most convenient for that type of investigation? And I'm convinced that those progenitors are called neural crest cells. Neural crest cells, they're often called the fourth germ layer, in addition to ectoderm, endoderm, and mesoderm, because they're so multipotent, they give rise to so many discrete fates in an embryo. And this progenitor type is extraordinarily counterintuitive, and typically people don't learn much about it during the university program. So neural crest cells initially are neuroepithelial cells. They originate at the neural plate border, these green bands. And when neural tube starts to fall, these neuroepithelial cells in these green bands, they undergo the process that is called EMT, epithelial mesenchymal transition, and they will delaminate, they will become mesenchymal amoeboid, and they will migrate everywhere in an embryo, and they will give rise to lots of different cell types, thousands of the cell types, some of which are non-ectomesenchymal, so there will be some neurons, some glial cells, chromatin neuroendocrine cells, melanocytes, our pigment cells, and there will be lots of ectomesenchymal fates that will give rise to a large portion of our face and the skeleton of our face, cartilage and bone. So basically, neural crest cells also make our face to a very large extent. So you see that they are very multipotent. They can do what mesoderm can do, what the ectoderm can do. Mesoderm makes basically mesodermal derivatives of cartilage, bone, muscles, and so on. And the ectoderm produces neural derivatives, and neural crest cells, they breach this kind of potency. So because they're so multipotent and because they do slightly different things in different places in the embryo, what you see here, so they have this system of biases stretching from anterior to posterior direction. It's great to study actually how they really execute that difference, right? And that's the depiction of the collective multipotency of the neural crest, that there are few cells that can be very early fate restricted, very few neural crest cells that just give rise to a couple of things. There will be really few cells that are absolutely multipotent, and they will realize their multipotency, fully giving rise to the entire spectrum of what is possible for them. But the majority of them, you see this bell-shaped distribution, right? The majority of them will interpret dynamic fate biases and generate the portions of what is available to them to different extent. And because there are many, they will still manage to build all the structures in the right numbers. It's a great mystery. How do they really achieve that? So previously, we tried to study it using single-cell information, single-cell transcriptomics. Many of you are familiar with the single-cell transcriptomics. You basically take a cell and you isolate the RNA from a single cell. You can do it in droplets, or you can do it in plates, like we did with SmartSeq deep sequencing approach. And then once you have the individually sequenced transcriptional information from every cell, you can cluster similar-looking transcriptomes and obtain this type of a tree. This is developmental tree, and here is the trunk neural crest. This is cranial, and actually you see different trajectories basically for neurogenesis, for mesenchymogenesis emerging actually from these places. So it's like an octopus, it's a lineage, right? And then you can actually ask, and when you navigate these bifurcations in the lineages, you can ask really which genes, which factors they will determine if the cells will go towards the red fate, for example, a neuron, or they will go towards the blue fate, will be like skeletogenic mesenchymal cell somewhere, right? So if you start back propagating the signal from the branches, you get this kind of like type of like jointly activating programs, and they fight each other and they compete with each other here in this pie. So there is a lot of fight, and the signals that come extrinsically from the niche and intrinsic cues, they try to support blue versus red fate. And two different programs, two gene regulatory networks co-assemble and they fight each other, and then one wins in a given cell, and that cell will go to the red branch, for example, right? So, and another cell might have a blue program winning, and it will go to the blue branch in that case, right? So, and you have a certain proportion, you have certain probability for every given cell to go to the blue branch or to go to the red branch, depending on the amount of support in the niche and amount of epigenetic inheritance like these cells have, which predisposes them to one choice over another. So yes, you can kind of like play between these two strategies to understand how cells execute these decisions and how they channel their multipotency into specific realization, and how these probabilities work, basically what influences these probabilities and how cells calculate the probabilities and decide something very binary. So we want much better system, because this takes a lot of assumption if we just, do these predictions based on gene expression. In a perfect world, we want to move from this kind of like, from this picture, what I showed you, to this one, where we can kind of label some progenitor cell, and then we can physically track the progeny of that cell and see in which branch that progeny will reside, and in which branches this progeny will not reside, right? And then we can track another progenitor and see how this different progenitor makes things different, right? So we can generate the map of the army of these progenitors with gene expression information that is barcoded, and we can connect the progenitor state with the output, and we can understand which molecular, cogs and wheels define what this early progenitor will give rise to, what is the meaningful macro heterogeneity in this specific individual progenitor cell. So technologically, we achieve it by performing ultrasound-guided microinjections of barcoding lentivirus in utero. So it's very simple system. Apparently, what we do, we just make or we get the library of the lentiviruses that can infect any embryonic cells. So what we put into these lentiviruses is the library of transcribed barcodes. So basically, these are just sequences of nucleotides in the DNA of the virus, A, T, C, G, whatever, like 10 to the power of 6. They're very diverse, very different, like barcodes, like real barcodes. And when the virus infects the cells, they're not just getting into the genome, they're also transcribed. So a lot of mRNA molecules are made. So when we sequence cells using single cell transcriptomics, we can get this information about the barcodes. So what we do with this suspension of viruses, we just microinject them into tiny embryos that are just 1 millimeter in length when they're still in the uterus inside of the mother in the mouse. So that's how this experiment looks like. And then we let the virus to infect basically all available cell types, including neural tube and the neural crest. Then we suture the mother back because it's a very non-invasive, it's not very invasive operation. So they're fine after the surgery because the capillary is very thin and it's very fine procedure. And we let them to develop the embryos much further. They can be bored even, like it's not a problem. And then we can collect different tissues and organs from these embryos, and we can unmix clonal information and cell types as the result of the single cell transcriptomics. So we can understand which phase, which cell types contain which barcodes, and then we can actually have a collection of different clones in different places, and we can try to understand what drives this diversity. Right. So we also work with cranial placos because they're very similar to the crest. They're also multipotent, and we need the second system to generate the data. So we don't want to be locked on one system. And cranial placos, they generate our sensory organs or components of the sensory organs. And they're also like very blurry in the beginning in terms of their multipotency potential. So these methods, they're really good to understand basically like how progenitors, different progenitors, they get kind of rooted to different progenies. Okay, so we do it with Gerhard Schlosser, and when we generate this first clonal information, we get the maps like this. So basically, this is binary, just showing you which fates are compatible with each other within clones. And that is actually how people generally look at clonal information when they do single cell transcriptomics. They want to have a map. If this fate can be correlated with this fate more often than it will correlate with this fate, right? So, and then they will do it pairwise to the other ones and so on. And this is really clumsy and bad way of analyzing this data. It's an informative way, nothing wrong with it, but we need something way, way better. And for that, we developed a new analytical technology based on artificial intelligence, basically word-like processing approach that we called clone2vec. Basically, what in essence it does, it is looking into the transcriptional embeddings of these single cells, what we have, and it's searching which cells belong to the same clone, and it will classify them. Basically, here you have like lots of red cells, they belong to the same clone. And they will appear similar to the cells that are green that also belong to a different clone. And then it will continue going on like this, wandering this embedding and seeing actually which clones occupy similar positions in the embeddings with similar diversity and proportions of cell types belonging to that clone. And then we can cluster similarly looking clones like red clone or green clone together. And the clones which are dissimilar, for example, like this blue, it's occupying mostly the central position in the embedding, unlike the green and the red, right? So we will get basically like some kind of clustering of the clones. So we'll have the cluster 1, cluster 2, cluster 3, and cluster 4. So we'll get 4 clusters. And that actually aggregation of this clonal information results in some kind of clonal typology, and this clonal typology becomes resistant to dropouts, and it's telling you way more information as compared to these, binary maps, which are absolutely ridiculous. I think this is way better system. So also we can do it in space, and we would like to do it in space. It just, I'm always bringing in this great example, this painting by George Seurat, right? So George Seurat was one of the founders of pointillism. And if you look here, all these human figures in this painting, they're made by the artist placing dots of different fixed colors in different proportions throughout the painting. Imagine that these different colors, they correspond to cell types. And these cell types, though being different, somewhere in space, they will be basically like they will be basically organized into clones. So this will be clone 1, for example, and somewhere here we will have clone 2, and somewhere here next to them we'll have clone 3, and so on, right? So basically the tissue of these humans, of us, is made by lots of clonal progenies placed proximally to each other, even though the cell types comprising these clones can be different, right? And still we have these anatomical transitions from the shoulder to the arm, right? And from the nose to the neck and so on. And our body is continual, right? So these clones, they demonstrate some continual axis of variation. Same in an embryo, some continual variation is responsible for the smooth changes in the anatomy. So it means also that clonal differences can be very subtle. Just proportions of cell types can vary a little bit here, a little bit there. Eventually, these differences will accumulate and you will get a different body part, basically consisting of the same cell types. Basically, my nose and my arm are made of literally same cell types. But they're positioned in different way. There are different proportions of them. Here we have like less cartilage, more bone, and so on. Here we have a lot of cartilage and less bone, and so on. We have a lot of epithelia and things like that. So how do we really solve this classification puzzle? This is really, really tough. And exactly this clone2vec comes in very nicely, because if we really work with transcriptional embeddings and we define the clone through learning the transcriptional context, like we learn, we can actually do, we can represent any word through its context. It's what any large language model does. But now we just apply it to the clonal information here. So we define the clone where the neighbors in this transcriptional space, basically cells that will be next to the cell of this clone. They will all be learned and calculated. Their transcription states will be calculated. There will be the context for the cell belonging to a specific clone. And this is now the whole clone, and this is the context for that clone. And now, after we do the learnings, at some point we have the representations, right? In basically in some kind of multidimensional space, basically where this information will be stored. And if we project that, we will get the clustering like in the single cell transcriptomics, where every cluster will contain the clones. Every dot here is a clone, lots of clones demonstrating similar distribution in transcriptional embeddings and similar type of proportional contribution in terms of fates, basically similar type of behavior. So we call them clonal clusters. And now we can ask which genes drive this variation of the clonal classes, why some of them are more discrete and some are more continual, and so on. So we can really go and do some kind of anatomy of these clonal classes, and we can extract gene expression programs and molecular mechanisms that make clones in different places at different time points more or less similar to each other. And this becomes very informative window into understanding embryonic development. How do we know that it works really well? We know it because we use a lot of in silico perturbations and simulations. When you work with such magic, you really have to control it. It means that we cannot even work probably like to great extent with natural cells. We want to create artificial cells. We want to simulate cells in the computer. So we don't simulate like a live cell or anything. We simulate the transcriptional state of a cell. We can make it into anything. We can make artificial neuron. We can make artificial muscle cell. We can make artificial abstract cell, and we can place it into transcriptional embedding. Here we have thousands of artificial cells being generated and placed into this transcriptional embedding. Basically, like every little axis of rotation here changes the transcriptional state of these basically artificial cells, and these are artificial cell types. So basically, this is the cell type 1, cell type 2, cell type 3, and you can see that in the center you have undifferentiated progenitor, and then here you have these axes of differentiation. So this will be the subtypes of the cell type 2. Basically, will be 4 subtypes like A, B, C, D, and so on, right? And then we do a bit of rotation, and now this guy is a bit different. So same progenitor, but it will navigate this trajectory instead of that trajectory which we had previous time. So now we have it a little bit different, and we go like this on and on and on, and we can actually invent different transcriptional topologies depending on like which processes and transcriptional states we would like to approximate. And you can see here we have like thousands of individual progenitor cells being simulated. But what if we introduce lots of dropouts like in a real experiment? This is the real this is not the real, this is simulated perfect situation where we have a readout from every simulated cell. But now we introduce dropouts. Let's say we only recover the fraction of cells. They will look like this. There will be one clone, this will be another clone. And you can still see that we are having these cross patterns, right? And here we have the circle pattern, right? Even if we do a lot of subsetting, we still see the circle. We can see it with our eyes, and the computer, the clone2Vec, of course, separates them really, really well in the transcriptional embedding. So we have this great degree of separation. So it works. We can push it to the limits. We can subset them way, way more at the point when human eye does not recognize the pattern any longer in these transcriptional embeddings. But if we have enough of these clones, if we have probably like hundreds of these clones, and they demonstrate still similar looking behavior, we can aggregate this information, and we can still recover the clonal information with a good degree of separation. Now this separation will not be as good as this separation, which you can see here, right? These clusters, they're very far apart. They're very discrete, very nicely spaced out. Here, they're not as nicely spaced out because there are lots of dropouts, but we still recover them. We can still return to some ground truth, and we can still see that we understand which clonal behaviors are different from other clonal behaviors. And this degree of robustness shows us that we can really take the true biological information from noisy readouts like single cell sequencing, we can still recover the clonal behaviors and we can study gene expression behind these clonal behaviors. I find it absolutely fascinating. So that's how the real data looks like, right? So that's how we observe these clonal transcriptomics niches. And here are the examples of individual clones that belong to one clonal cluster. This is the real transcriptional embedding. This is the entire mesenchyme from the embryo at 15.5. This is mouse embryo. So these are all mesenchymal cells. Some of them will give rise to cartilage and bone. Some of them will give rise to smooth muscle, dermis, other types of soft connective tissue. And you can see that basically when we sequence them out, the clones occupy kind of like these spaces like this, right? And they appear a bit similar, right, to each other. So all these clones, they show certain pattern, right? So, and there will be thousands and thousands more of these clones, right? So they probably will make what we call the clonal cluster, right? And this is something very good because if we would use RNA velocity here, another method to infer directionality in single cell data sets, it would point out in all directions. It would say like the progenitors probably reside somewhere here and they can differentiate everywhere. But do they really differentiate everywhere? If progenitors are sitting here, these progenitors that give rise to these clones, to this clonotype, they do not go here, they do not go here, they do not go here, and they do not go here. Instead, they cover this upper part. Instead, they cover this branch, this part of embedding. So these progenitors residing here that give rise to this, they are already biased, they're already tilted. They ignore these lower parts and the left part of this embedding. So it means that you cannot rely on velocity for understanding what exact progenitors can do and what they cannot do, but you can rely on the clonal clusters. So these are the densities of the clonal clusters, of different clonal clusters plotted. And you can see that clonal cluster 1, for example, is very different in its multipotency realization as compared to clonal cluster 8, even though they start from somewhat this area where we have these progenitors. So all these clonal clusters start from approximately same space, but they do very different things. And why? Because basically progenitors would respond to different spatial and positional cues in different spaces or locations of the embryo. They will try to build different anatomy. And for this, they need to create different proportions of different cell types from the same multipotent progenitors. It's like a clay. You can take a clay, right? And you can make a cup of this clay. You can make a bottle from the same clay. You can make a human sculpture from the same clay. These are three types of very different objects that you will make as a creator from the same clay. And here, nature takes this same type of mesenchymal progenitors and makes different things and different body parts come to existence. The hand, the leg, the nose, the ear, and so on. This is how this is achieved. And we want to know about molecular signals, how it's done, which are driving these differences. So for this, we can take two clonal clusters. We can take the blue and we can take the red. They're next to each other on this embedding. And we can say, can we really explain molecularly the very fine difference between these two clonal clusters? The true difference is that the red one gives rise to this branch, which is the branch to the bone. And the blue one doesn't do it. The blue one does everything. That's similar to the red, but red is exclusive. This type of red branch is something the blue is incapable of doing. And basically the trajectory can be approximated like this. And here's a straight line in this part, right? So now we can actually take these two trajectories and we have the bifurcation in one of them. We can overlay them and we can actually, we can ask what's going on in this area where we have this bifurcation. What's the difference? And this is the difference. These are specific genes. This is the heat map for the genes. And these genes introduce the difference into one clonal cluster, permitting this bifurcation. So now we know the genes, we can target them, we can try to comprehend it at the level of the molecular mechanism, right? And then we can go into these genes and we can try to find where they are in the embryo using spatial transcriptomics, some sort of multiplex in situ hybridization. And we can see that in case of the clonal cluster that gives rise to the bone, it's tilted to rib cage. And the rib cage produces the bone. And the spine does not produce the bone at E13.5 because the spine will ossify much later. as compared to the ribs. So basically, like the red trajectory, this is ribs. And this is the spine, and the ribs are very close to the spine. That's why these two clonal clusters are next to each other. And specific genes, they will respond to the environment where we have ribs, and they will launch the ossification program here. And it means that, in fact, this transcriptional embedding, where we have different clonal information and all these branches can be unmixed in different locations in an embryo where things go different ways, right? And it tells us that these embeddings, both transcriptional and clonal, they have this polarization, they have these axes of polarization. And these axes of polarization, they exist because there is a positional information like Hoax code in the embryos, and not only Hoax code, some other positional information that we fully comprehend today, and we can fish out through this analysis that helps us to understand which genes are driving certain axes of variation, how varying clonal clusters relate to this gene expression. Here we just go for Hoax genes because we know they're going to be involved. And this is just an intuitive thing to do, but you can be absolutely unbiased in this investigation. Right? So if you do it in an unbiased way, you start seeing the genes like POSTON, like ZIC1. You can start seeing actually something like SHOX2, MACE1, TBX3, OS1. So you start seeing genes that are not Hoax genes that were previously not associated with positional information, but now you recover them as genes associated with positional information, with niche information. And it helps you to discover and understand positional information in a new way at the level of basically microscale, because Hoax genes, they will define much larger domains, but we still fully understand how positional information translates into some specific shaping and differentiation outcomes at the microscales. And this is the way to go. This is the way to study this using these embeddings. It means that if we look at the cell types, right? If we look at any cell type in developing embryo, the differentiation trajectory in single cell embedding will look something like this, right? But depending on the position, there will be some kind of auxiliary positional program that will influence when the cell will differentiate, will define the pace of differentiation, will define the subtype of differentiation, and so on. And will be the ground basic program that is responsible for the fundamental aspects of the differentiation, not a kind of specific space and timing kind of like whatever, like controlled aspects, which are very fine aspects, but some kind of like a major thing. So now you can unmix them using this analysis and comparisons of clonal clusters. You can basically distill this program into two: basic and accessory program that makes subspecializations and executes things very precisely in different parts of an embryo. So, and it really expands our capacity to understand the embryonic development. So that was about the descriptive part. So that's how we basically predict and see things, right? But we need to prove that what we predict, what we think, has functional consequence, that it works mechanistically. And here we have to do it via the perturbations. And we want to do the perturbations in a multiplexed way at clonal level. It means that every clone we want to perturb differently, and we want to have a collection of thousands of these clones from a single embryo. So in this case, we wanted to understand which molecular signals that act on progenitors, how they will shape the clones. So we took the data, the single-cell transcriptomics data of the neurocrest, and we basically predicted which signals will act on receptors prior to bifurcations. So these are all receptors and signaling molecules. And we introduced not only the clonal barcodes, but also Cas9 CRISPR guides that can kind of cut out these receptors in clones which are getting the barcode and which are getting targeted, right? So now we know which clones do we have, and we know which receptor is missing in these clones during the neurocrest development. We sequence them out, we make clonal embeddings. So these are clonal embeddings, and versus the control experiment where we don't have CRISPR guides, we search for novel clonal clusters like those which emerge. These are abnormal patterns of clonal behavior that do not exist in control. So when we see these novel clonal clusters forming, we know that cells, when something is missing, progenitor cells, they create clones that don't look like natural. They have collections of fates that don't occur in natural way. Not because they will contain some kind of new cell types, probably, but because they will have abnormal proportions of normal cell types, and that will render these clones already wrong or different, right? So in this case, those brown abnormal clones in clonal classes, they correspond to the loss of patch 1, and patch 1 loss means activation of sonic hedgehog pathway. So these are like sonic activated clones all the time, and they generate these structures. And we have many other examples. These effects, they can depend on the position because that's how these abnormal clonal clusters look like on the embeddings, right? This is mesenchymal and this is nervous system. And you can see that in the nervous system, they start from progenitors and they cannot finish the differentiation into neurons, basically. This is really, really ridiculous thing that they get arrested as progenitors. And funny part is that this effect has this two-hump structure, which means that it depends on the Hox genes, on the Hox core. So this is somewhat depending on the positional information. So in some contexts, it works much stronger. And in some contexts, it's very weak, almost no effect. So there is something that kind of confounds it. And it's interesting. What can it be? So it gives you new information about the molecular mechanisms of how sonic is shaping actually the fate selection and the structure of the clones during the process of embryonic development, right? So, and we can actually expand it further. We can go into the neurocrafts, which we love, and we can describe the system of their natural biases, right? When they migrate, when they exit the dorsal neural tube and they start to migrate in an embryo, they're thinking, what are we going to give rise to? And every individual cell will ask this question again and again and again and again, and it will get individual answer every time. So there are groups of cells with enteric nervous system bias. Those guys, they'll say, like, yeah, we will go into the gut and we will make these neurons into the gut so the gut will become motile. And some others, they will migrate slightly further away. They will be subjected to different signals. And they'll say, yeah, we would like to give rise to the sensory neurons that gives us information about the temperature or touch or pain, right? This is very important contribution. And these guys will be, yeah, we are going to do this part. So there will be actually individual progenitors that will be biased in these different ways. And now, if we introduce these Cas9 CRISPR guides, can we perturb those biases? And the answer is yes. Depending on which receptor we cancel here, we're getting slightly different biasing result. For instance, if we start to disturb frizzles, the wind system, right? We are getting enteric and sympathetic neuronal bias much less. So basically, these cells with these perturbed receptors, they don't want to become neurons. They want to become glial cells or sensory neurons, not autonomic neurons. So we start to reshape their proportions. We start to do this influence, and we can validate our predictions in that way, which is quite cool, right? Because if you can do this type of confirmations, your system starts to be really testable, right? That's the core idea of this type of experimentation. And what we can also do, we can ask very cool questions about compensatory mechanisms in the clones. We know, for example, that embryos, they regenerate really well, right? Embryos regenerate much better than adult bodies. Not only because they're small, but because the mechanisms providing for robustness are way more active in an embryo. It's a system of crafting the body that must be resistant to errors. It must have some kind of potential and mechanisms to correct the errors, to detect them, and to rebuild things, right? How do they do this? Which molecules are responsible for this? How do we study this? And for instance, here I will give one example. It is very well known that if mothers, when they are pregnant during the first couple of weeks of pregnancy, if they drink a lot of alcohol, there is a high chance for congenital defects, specifically that result from the neurocrest lineage, because neurocrest cells, they actually they poorly tolerate alcohol. So basically, alcohol is poison for the neural crest, and actually, a certain proportion of the neural crest cells will die if we have a lot of alcohol in the system of this embryo. So, but the remaining cells that will survive, they will try to cross compensate, and they can achieve certain degree of success depending on how much alcohol was put into the system or how many cells died. So there are some limits to these compensations, right? And in the lab, we have this fetal alcohol mouse model. We have this fetal alcohol syndrome model, which we can reproduce, and we can actually add alcohol into these embryos. And we can kill a portion of the neural crest cells, and we can see if the remaining cells will start being more kind of multipotent in terms of how they execute their general multipotency as potency, right? And whether they will help to rebuild the structure, even though some of their neighbors died out and their numbers are not sufficient. So what will be the compensation here? We are still in the middle of these experiments. I don't have much results to show you. I can just say that we do see some clonal compensation events in our real data, and it all looks super exciting. Also, what we can do, we can actually try to understand how progenitors gradually lose this competence over time. We can inject the virus now at two different time points. At E7.5, when clones are still early and progenitors are more multipotent, and just 24 hours later at 8.5. And you can see here like the results of this experiment. So the blue is blue, orange is orange, and you can see that basically the blue clones that they occupy way more space, they're more multipotent. So basically in this area you almost don't see anything orange, which means that orange cells already got fate restricted just during these 24 hours. They will not generate these clonal structures that would account for this part of the clonal embedding. And we could, by studying these clonal embeddings, we could predict that there will be two waves of the neural crest migration. Will be this early wave that will give rise to the skeleton of the face, and there will be this late wave that will give rise to the nervous system of the head, trigeminal ganglion, and all the glial neural structures. So they will come in some sort of a sequence, and the early one will be more multipotent than the late one, which will give rise only to the nervous system components, predominantly to the nervous system components, not exclusively, but predominantly. And when we validated with lineage tracing, that's exactly what we see. We have just, small shift in injection of tamoxifen, and here we have the full face label. This is the mouth. This is the mouth part. This is the eye here, the forming eye, right? This is the jaw. And you see this whole face being traced, including the ganglion, which sits here. And if we just do it slightly later, we see only the ganglion with its kind of like branches being traced. And actually, this is the eye, and this is the mouth. And all these things appear largely untraced. So there is a big distinction between these two things, like what we see here. And now we know that when neural crest cells, they build different parts of the face, skeletal or neural, they do it in some sequence. And the clones that are responsible for this, they lose some part of the competence, or they just choose a different clonal, outputs, different phase to which they will give rise to in a new context, in a new time, right? And these embeddings, they help us to discover such things. So they become very important instruments for basically predicting such situations in an embryos. And now we move beyond that to the next step. So this is something basically very new. So before we could recover the clones only by sequencing cell transcriptomes, we had to dissociate the embryo into suspension of the cells. And now we are creating spatial transcriptomics compatible barcodes. So now we can see the clones in space on the sections of the embryo. And this is really, really powerful. Basically, we see thousands of the clones. And if we zoom in into the nervous system, into the hindbrain, we will see these beautiful columns, right? And these columns look like this, like you see them here, high magnification in the spatial data. And it really corresponds to the ground truth, what we know about the development of the nervous system. So now we preserve the positional information and we preserve transcriptional information. So now we know which signaling factors they act on the cells in this exact specific location, right? And how it differs, for example, from clone 2, from this clone here, right? So now we have the access to the spatial information, which makes the whole analysis way more powerful. And we are creating now the very first atlas of this special clonal embryonic development in mouse. We are looking forward to see these results. We don't have them fully analyzed yet, but it's a very promising strategy. So I will now just list our delicious questions because, that's what we want to learn. We want to understand how can clonal variation help us to understand cell fate control and development in general. Like when we look at the level of individual progenitors, we would like to understand how cell type proportions are set, how the probabilities of transitions for individual progenitors are defined, which molecular mechanisms are responsible for this. We would like to understand how signals and cell-cell communication plays a role in this process, in this collective multipotency and in robustness when we observe these clonal compensation events. We would like to understand how positions and the timing introduce fate biases into fate decisions. And we, of course, need to build new clonal biology tools to detect clones and to analyze clones, like clone-to-back, these transcriptional embeddings, or even like transcriptionally, like transcriptional barcodes, the clonal barcodes that we can pick up and detect in space, right? So basically, we would like to record a lot of new information, including clonal information in space. And this is still in the very beginning. So this is the future of the field. And we really look forward to make a lot of progress there. And with this, I will thank my lab and all the people who played a major role in the development of this project. It was mostly Sergei Saev, Alek Eriksson, of course, Peter Harchenko, Johan Bergstrom, and Louis Faure, who is now a postdoc in the United States. So yes, we will keep going. And please, we have time for questions now. Ask all your questions. I hope my lecture was comprehensive enough. Thank you very much.